inviting me to participate in this nice session. I also need to thank, or want to thank, and acknowledge very quickly the number of people who make it possible for me to stand here and tell you the stories and ideas I'm going to tell you about. I'm going to give you an evolutionary context for thinking about life in ice and why particularly to focus on Arctic winter sea ice. I'll walk you through a couple of what I think are wondrous adaptations to life in ice, and finally tell you what I mean by evolutionary playground. And if there's time, I'll take you to the coldest sea ice habitat. So here's my favorite quote from Darwin. It's perhaps the most famous one that ends his book. And many of us think of him as this wise, elderly fellow, but his discoveries of the most wondrous and beautiful forms of life came as a young man on this voyage of the Beagle, and it was right after he returned from that trip that he drew this first tree of life, which is quite remarkable when we compare it to the tree of life we have today, which is a phylogenetic tree of life, of course, based on genes and sequences of DNA, where every length of every line on this curve has a specific quantitative and evolutionary meaning. Darwin did not know the term gene or DNA and barely knew about the existence of microorganisms. In fact, didn't know what to do with them. That word doesn't appear in the first issue of his book. So I have the challenge of bringing microorganisms to wondrous diversity life for you. Here is the animal world, animal plant world that Darwin had to deal with and that we've heard a lot about today, based on morphology, based on what you can see with your eyes, what you can see in the classroom, what you can see in the field. What I need to do for you, which an earlier speaker did for you for the deep ocean and how it is the dominant part of our biosphere, we are in fact a microbial world on this planet today. Most of the genetic diversity is in the microorganism space in life, not what we can see. And that's hard to take in sometimes, especially since they all look alike. They're all tiny 1 micrometer sized bacteria and archaea that are very difficult to differentiate morphologically, if not impossible. So of course in modern times, we've developed genetic techniques to make them light up according to who they are and all this sort of thing. But where they excel, where their diversity comes from, is in all the wondrous things they can do with their enzymes and their metabolisms, things that plants and animals can't even think of doing, extreme environments they can live in that plants and animals can go nowhere near. And the area of temperature is what I'll talk about today. Rooting the tree of life are heat-loving microorganisms, some of which live in very extreme temperatures. Up to 122 is the limit in pure culture in life in the laboratory today, to the more recently evolved cold-loving ones, and even ice-loving microbes, and that's where I'll take us today. So this is a different kind of voyage than Darwin was able to make. I'll take you into the Arctic Ocean here, and if my film works for you, let me first set the stage. Here's Greenland, which is covered with glacial ice, freshwater ice, that's not moving except when it calves and with global climate change issues, but a set amount of freshwater ice on the continent there. But what you're seeing here is sea ice. Every year, the surface of the seawater freezes and circulates in the Arctic Ocean. Some of it survives several years and before it melts out. But there's an annual cycle here. So this is the year 2003 in October. This is the minimum extent of the sea ice cap. So the blue is open water. That's Russia up there. Here's Alaska. This is the Canadian archipelago. And coming down through Greenland, here's the ice cap. Let's see. So we're going to watch the time here. We're going into the dead of winter. You can see the ice cap fill out all the space there is, but it never stops. It's a very dynamic environment. It's cracking. There are hemispheric scale cracks in the ice here. Very dynamic. We'll come into springtime. You'll begin to see it pull away from the land masses. You'll see open water here. Lots of atmospheric storm activity. Very exciting place to visit, let me tell you. till we get to, we're about to end to the minimum extent again in the year 2003. Okay. So 10 or 11 years ago, I was on an icebreaker working right here in open water. I hadn't yet got fixated with ice. But while I was there on this Canadian icebreaker, the Amundsen, which is quite a wonderful ship, I was distracted by the ice. And when I got home that year to my office, Science magazine had images from the NASA Galileo mission coming in. So there was a space mission that flew past the moon Europa to Jupiter and sent back these spectacular images, which to me and to a lot of people look like, oh, cracking ice, just like and you can see wonderful features on the surface where you have slip functions here. You have areas that look like they could be warmer, a dynamic situation here. So the first other ocean found in our solar system here, which volumetrically is twice the volume of water we have on this planet, or that's the best. It's actually today it's n equals 8. There are probably 8 other oceans in our solar system, but that's another story. So I joined colleagues at the University of Washington, and we established an astrobiology program where my role is to study life in cold ice. And the reason is that the candidates for the possibility of life out there are all on planets or moons that have frozen surfaces. So if we're ever going to be able to sample any of them, we're going to be getting ice. So I figure we need to look back on Earth, where we do still have some very cold temperatures, and learn as much as possible about life in ice. Aside from the astrobiological issue, this whole retooling and redirectioning of my own research career is introducing me and my students, and I hope all of you, to the wonders of life on this planet. Forget the rest of it if you're not into that. So this is the year 2007, which some of you may know was a record year of sea ice loss. Here we are again. Now you can see the Greenland ice cap, Alaska. So this is the dead of winter now with the maximum ice extent, which doesn't tell you about the thickness of the ice. The ice is actually thinning. And what you're going to see now coming into September, which is the minimum extent month of all time, is this great loss of sea ice here. This loss of real estate here, if you will, is twice the, you could put 2 Texases in there. So it's a lot of real estate that we're losing. It's an endangered habitat if you're interested in knowing about life in ice. So there's some urgency here, or I feel some urgency in my lifetime and my students' lifetime to learn as much as we can. We first went into the Arctic in the wintertime north of Barrow, Alaska, some time ago without much fancy equipment. We used the research vessel sled. It's an Inuit sled, so we were taking advantage of Inuit knowledge and technology here. You have to have intrepid graduate students willing to work in very severe conditions. You have to be kind of crazy yourself. And what we were after were cores of sea ice into this ice. We're standing on ice and you're seeing snow everywhere. It's near -2 degrees at the bottom of the ice where it interfaces with the ocean, which of course is liquid. The freezing point of seawater is about -1.9. Then you get ice, and up near the atmosphere you get much colder ice, so we're after the cold ice. Here's a typical ice core that you pull up in springtime, and here's a rare case where you can visualize the presence of microorganisms because there's so many of them. These are ice algae giving a pigmented look to the core here, and those algae can be massively productive. Now we're looking on the underside of the sea ice, and they can be like a forest draping from the ice meters long in some cases. This is a famous picture from Igor Melnikov's book on sea ice. They support a whole ecosystem, including our favorite charismatic megafauna. But this is not where I'm headed. I need to take you microscopically into the ice, which is a porous matrix. So here are the caves, if you will, the cave equivalents of sea ice. And at this stage, on the millimeter scale now, We still have a lot of predators. We have a lot of diverse higher forms of life here. The bacteria, the little dots here, aren't even showing up there. So I'm still at the wrong scale for microbes. Too big. And I'm really not interested in that warm ice. I want to get up to the cold ice. And something very interesting in sea ice happens at about -5 degrees. As it gets colder and colder, the ice becomes impermeable. So nothing can leave it anymore. So whatever organisms are in that ice are now trapped, and they're going to go through the winter time and experience very severe conditions. And they have no predators, because as you'll see in a minute, the pore spaces in there get so small, there's only room for bacteria and archaea. You can teach your students very easily about this porosity environment of sea ice with a little, or in your kitchen with your kids. This is freshwater ice made in an ice cube from tap water, and this is made from salt water, table salt dissolved in salt water and frozen, with food coloring dripped on it. The freshwater ice, the food coloring slides off of it. It doesn't soak it up. It's not a porous ice. No habitats in there. Sea ice is highly porous, soaks up the dye. It's evidence of the porous space in there, habitats for microbes. Let's look at that a bit more formally. These are panels of sea ice, 30 millimeter square panels, starting with relatively warm ice, where the white is the solid form of H2O, and the black is the liquid salty brine, kept liquid because salt depresses the freezing point of seawater. Okay, as you drop the temperature on that ice, you shrink up all those pore spaces. And as you get below -5, there's no expelling of any of this salt. So what happens is those pore spaces shrink up, and you have what we call a brine concentrating effect. So because nothing can leave the ice, everything gets concentrated into those pores. The salt concentration zooms up. The number of microbes in there become more concentrated. Anything that was in there before becomes more concentrated. It becomes a very exciting place. Saltier than what we use to can food and prevent microbes from growing, and it's very cold. The other problem is that we are still at the millimeter scale, and we're interested in microorganisms that are 1,000 times smaller than these pores. So what we tried to do, what we did succeed in doing, was figure out how to look into sea ice at the micrometer scale. Again, it takes a graduate student prepared to spend part of her life in a freezer lab with a factory-modified microscope, computer imagery analysis. What she figured out how to do is take a slice of natural sea ice, microtome it. She figured out how to add a DNA-specific stain to the surface of it so that it would move into those open pores and stain whatever might be in there. The next image that I'll show you is without any stain, but it was the first image ever looking into sea ice at this scale. We were terribly excited. It's not a very dramatic picture, but it was very exciting for us. What you're seeing here is the solid phase of ice, and these are brine pores. And the exciting thing is that they are connected. There's a liquid network even at -20 degrees in sea ice. And this was not appreciated before anybody actually looked into the ice at this scale. This is really good news for life. There can be exchange between the pores. It's not a closed system, it's an open system. Excuse me, let me grab some water. Here also with no staining are a pair of diatoms, actually 2 pair of diatoms, so sea ice algae. No staining, you're seeing their green fluorescing chloroplasts, happy, intact. They're not photosynthesizing because this is the dark of winter, there's not enough light, but they're surviving through the winter just fine, morphologically anyway. Now if we switch to a DNA-specific stain, then you can see the 2 pair of diatoms, but now you can also see for the first time the bacteria. And there are lots of them. I haven't put all the arrows in there. We used another stain that picks up sugar molecules. And we quickly discovered that all the sea ice algae in there are coated up with something we call EPS, extracellular polysaccharide substances. sugars, basically. It's equivalent to mucus that we coat ourselves up with when we get cold. And it functions as an antifreeze. It depresses the freezing point of this brine even further than the salt does. It's a natural adaptation to living in ice. And it's in every pore we saw. This sugary, gelatinous material is in every pore, whether there's an organism visible in there or not, and whether you stain it or not. This is unstained, but I think you can see the gelatinous stuff that's in there. These organisms are smart. And this is an example, a rare example of biology altering physics. So these panels on the left are close-ups of sea ice without any of this EPS in it. This is sea ice made with the EPS in it. You can see the difference in the pore habitat. Circular, closed pores, very boring. Interconnected, increased surface area, and improved habitat for life, and the microbes did it themselves with their extracellular releases. They changed the physics of their habitat. Back to our very cold ice and bacteria. So now we're at the top of this ice. It's called a triple point juncture where 3 ice crystals are together, and there's just a little bit of space in there for a little bit of liquid brine, and we still see bacteria in them. And my favorite shot, I know these are not morphologically beautiful, but this is minus 15 degrees, and here we have a bacterium caught in the act of dividing. You can quibble with this as evidence for dividing, and some of our reviewers did. How do you know, Jody, that that wasn't dividing before it was frozen in and then it just stopped? Well, there's other evidence from other labs and from us as well. Minus 15 degrees, we do have bacteria that can grow under those conditions. So that's the lower temperature limit for life at the moment based on reproduction of a bacterium. We've also used molecular techniques to demonstrate that at the very coldest temperatures in those first cores we got, all of the active bacteria that we could score as active were attached to something. They were embedded in EPS. And to make a long story short, because a lot of other people have done work on this now, not only sea ice algae produce all this goop, but bacteria coat themselves up as well. It seems to be a universal adaptation to surviving, even living in these ice formations. This kind of data came from a single core in March. Well, replicate cores, but a snapshot of what's happening in March. We wanted to know what happens through the whole winter season. So we got a little more serious. We generated an overwintering project using the Canadian icebreaker, and we froze in here north of Canada. Alaska's over here. Very unusual experience to freeze a ship in for 6 months. Captains don't know what to do with themselves because they're trained to navigate a ship, and the ship didn't move for 6 months. So we had a different captain. This is Noel Green, who is an Inuit wildlife observer on board with us, with his rifle to protect us from our own predators. And he became our captain of the ice because he was uniquely adapted to this cold environment, as we've heard humans can be to extreme environments. But my question is the ice he's standing on, is it a museum? Are these things contained in there through the wintertime, not doing much? Or is it actually an evolutionary playground? So what do I mean by that? This is what I don't mean by that. These are human playgrounds where you release energy, you get through the workday, you have international exchange. A lot of good things happen with these sort of playgrounds, but at the end of the day, even at the end of the overwintering expedition, we are all still genetically intact. We haven't changed. I'm after, is there something going on in the ice that allows for evolution to go forward in a winter season? This film is not going to play, but I think I need to move on anyway. So we looked into ice through the winter. This is the thickness of the ice when we arrived on January 10th. So ice freezes from the surface and grows from the bottom. And this is a temperature contour, so the warm temperatures are on the bottom and the cold temperatures at the top. And we sampled regularly through the coldest part of the ice, so we were able to get colder temperatures this time than we did before, and to track what happens to the microbes through the whole winter season. This is bacterial abundance through the winter, so over the course of 3 months. The general impression is that they are persisting pretty well. If you look closely, we have some significant decreases in numbers, up to about 40% of the numbers. But since we've got on the order of 100,000 bacteria per milliliter of ice, we still have lots of bacteria persisting through there. And the other thing that we noticed was that EPS, this sugary mucus stuff, is increasing throughout the winter. And this is in the part of the ice where there are only bacteria and archaea present. No sea ice algae to do this work for them. And so they're doing this work. This is their strategy for surviving. But even with all this work to survive, and these survive quite nicely with the highest production of EPS, we do have some losses. So what's causing those losses? And we could discuss lots of possibilities, but the one I'm going to throw at you is that there are indeed predators in that ice. They just aren't bigger than the bacteria. They are smaller than the bacteria, viruses that attack bacteria. This is a cartoon of T4 phage that attacks E. coli from the human gut. We know everything there is to know about this. Not me personally, but the scientific community knows everything there is to know about this. This isn't playful. We know the proteins that make up this body here. We know the DNA structure here. We know the landing fibers. We know everything. You can see the virus is actually attacking an E. coli cell here, and my favorite part, I should be using the pointer, I suppose. You can see a virus injecting its DNA into the cell. Okay, a couple of things can happen there. The DNA can enter the cell, take over, reproduce itself, and kill the bacterial host. That's lysis, cell lysis. That's obviously bad news evolutionarily, although it's a selective process, isn't it? Or that DNA that enters the cell can benignly join with the bacterial genome and possibly bring in a new gene that it picked up from its former host, which we call horizontal gene transfer. So you can have some bad news or you can have some good news. And the question I'm posing and wanting you to think about is if it's possible for this to be happening under very extreme conditions and to explain a rapid adaptive response. So these experiments are very hard to do. I'll just show you an example of one where we looked at numbers of viruses per mil of brine from very cold sea ice, numbers of bacteria over incubation time in the shipboard laboratory. So we were in the field, but in the laboratory in the field. These were the most extreme conditions we could create stably in the lab. And the main point here is that it's not a static system. We have bacterial increases and decreases. We have viral production and viral decreases. There's stuff going on here. When we calculated the density of the viruses that must have been in the brine pockets originally, they are very high, 10 to the 8th viruses per mil of that brine. Doesn't get any higher than that. That's because of the brine concentrating factor, remember. And if you calculate the contact rate between a virus and a bacterium, when they're all concentrated into these brines, it's nearly 600 times what would be experienced in seawater itself. So just by virtue of freezing seawater, you are concentrating things, bringing things together, evolutionary agents together to make some things happen. To progress beyond what we can do in the field, we have laboratory models. So we have our favorite cold-loving bacterium named after Rita Colwell, Colwellia cycloerythraea. It holds records for doing just about everything at low temperature. I don't have time to tell you all of its stories, but we also have a virus that attacks it, and we have reproduced what we measured with the field samples, so we know that this can go forward. We have the whole genome sequence from our bacterium, and I've just picked out a few bits of evidence from there. First of all, this organism holds the record for the number of genes involved in releasing things, producing and releasing things, like antifreeze, like EPS, as a part of the general strategies for surviving in ice. And there are some archaeal genes present in here. Bacteria and archaea are two totally separate domains that is evidence, or hints anyway, of horizontal gene transfer. And there are two complete sets of genes of filamentous viruses sitting in that genome. Again, the potential for horizontal gene transfer to have occurred when those viruses came into the genome. And this is not my work, but another group's work, where they have plotted all of the genes that have been sequenced from an Antarctic bacterium that's also cold-loving, found from ice. And if you look at the gold-colored genes, they are all virally derived. It's about 25% of the genes can be scored as having been introduced by horizontal gene transfer. And the third genome sequence that's now available, this guy, Cycloflexus torcus, also an Antarctic strain, also has a significant infiltration of genes by the agent of viruses. So here's what I mean by an evolutionary playground. I mean this pore space in very cold winter sea ice where you have a bacterium that's coated itself up so it can survive there, even altered its environment so it's more favorable to it, but there are plenty of viruses in there to either take it out or give it something to help it even further. I've got a little bit of time to go to the coldest possible sea ice. So we've been back to the Arctic recently, another overwintering expedition. Here's where we had frozen in before, but we froze in and didn't move. So we watched stable ice through time. This time we kept the ship mobile. Working in leads. So leads are open lines in the water where the ice floes are breaking apart. And when in wintertime, whether the lead is broken by the ship or naturally occurring, soon as that open water occurs, it freezes because the atmosphere is very cold. It's -20, -30, -40 degrees, depending on where you are. Here's a satellite image showing you the vast number of leads and the extent of them where we were traveling with the ship. So here's a natural lead that has formed, broken in the ice. And if you can see this sort of hazy stuff in there, that's the new ice that's just frozen. Football field sizes here we're talking about, kilometers off into the distance. And if you look up close on that new ice, we have frost flowers. So this is a scale of maybe 2 or 3 centimeters. So these frost flowers are beautiful. They've been observed by Arctic explorers and Antarctic explorers forever, but as items of beauty. Physicists are very interested in them today because there's so much ice crystal surface here that they reflect a lot of light. They affect the albedo, or the fate of radiation coming into the planet. Nobody has looked at them as a habitat for life. So we did that. This is Marcela Everett Sarmiento from Colombia, who had never experienced cold temperatures at all. And she is the first to have sampled, what did I do? Well, what did I do? She is the first to have sampled frost flowers as a microbial habitat. Do I need help here? I'll keep going in the interest of time. These are close-ups of winter frost flowers. We don't have much light to see them by. Sorry. This is another student, Min, who grew her own frost flowers, but they were not for sampling. Okay. The gist of this is that our preliminary analyses showed the highest concentrations of bacteria or viruses that we had seen in any sea ice at all. And we've learned how to grow the frost flowers in a laboratory freezer, so we can do controlled experiments now. And what we can see is that bacteria in the source solution that you go to freeze travel up through the new ice in the brine channels and are expelled up to the surface of the ice where they freeze because the air above is much colder. And so you have the concentration of bacteria within the sea ice pores concentrating even further on the surface as they freeze. And they're traveling, also traveling with the bacteria are very high densities of viruses. What we don't know yet, and what you can imagine we're going to try to find out in the coming years, is if they're doing anything at minus 26, minus 30 degrees, or if this really is the dead end now, and then we're hitting the boundary for what they can, where they can act or do anything. But I'm the eternal optimist and think that we don't know the end of evolutionary patterns yet, and extreme environments have no meaning to these microbes. So I'm looking for a positive answer, but that's the motivation. Of course, we'll be rigorous in how we deal with this. I didn't get to show, if you can, there's just one last image that shows you a field of frost flowers. Yeah, if you can just go to the end. All the way. That's where I was, but I think I've told you all these stories now. Growing frost flowers, finding these very high concentrations. Here's the little scenario. The question is this a dynamic system microbially, evolutionarily, or not? And Darwin had it right, and we still have a lot to learn about wondrous things on this planet, whether we worry about other planets yet or not. Thanks very much.